Hello anatomist, here's your lab prep video for the human digestive, respiratory, and lymphatic systems. First, we're going to cover the digestive system. One of the things you need to be familiar with is the difference between organs that are part of the alimentary canal, or GI tract, and the accessory organs. The alimentary canal is composed of organs that are in the tube that leads from the mouth to the anus. So the mouth, the esophagus, stomach, small and large intestines, all of these are part of the alimentary canal, or GI tract. Accessory organs are also really important in digestion, for example, the gallbladder, the pancreas, and the liver, but they are not part of the tube. So you have alimentary canal organs and accessory organs. Both types are going to be important in the digestive system. We'll start at the top with the mouth and the salivary glands. So in the oral cavity, you have your teeth, your tongue, and other structures that help with digestion. And then you have salivary glands. So the really big one just near your ears is the parotid gland. You do have right and left parotid glands. Under the tongue is going to be your sublingual gland. And then down here by your mandible will be your submandibular gland. And you have right and left for all of these, so don't forget to put that down on your answers. If you take a look at the salivary glands under the microscope, here's what a couple of them look like. The submandibular gland, you can see some serous cells here. They stain a little bit more darkly, and some mucus cells that are more lightly staining over here. You'll also see several ducts. That saliva needs to be taken to the mouth, and the ducts will transport it there. If you look at different types of salivary glands, over here you can see the parotid salivary gland. It may look a little bit different, and this is because different salivary glands have different proportions of serous cells and mucus cells. Because the parotid salivary gland is composed of many more serous cells and not as many mucus cells, it has a different look to it. For lab, you will only need to be able to identify these as salivary gland. You will not need to identify the specific name of the salivary gland, but that's only for histology. On a model, you will need to identify the specific names of the salivary glands. Before we move on, I'd like to cover the layers of the GI tract. Similar to blood vessels, you have different tunics in the GI tract, or the alimentary canal, and these are consistent throughout the alimentary canal. So the innermost layer we see here is the mucosa, and the mucosa has a couple of subsections here. The innermost layer, you have stratified squamous epithelium is shown here, so it must be the esophagus, but that will change depending on what region you're in, what organ you're in. So it will be simple columnar epithelium when we're looking at the stomach, for example. But you have some kind of epithelium lining the inner side. Then you have lamina propria. Lamina propria is just the name for the connective tissue that is present in a mucous membrane. And the digestive tract has a mucous membrane on it, so this is the lamina propria. Beyond that, you see this very, very thin layer of muscle. This is the muscularis mucosa, just a very thin layer of muscle. But all three of these little layers are all part of the mucosa. And then as we move more superficial, here we have the submucosa. So submucosa is outside the mucosa, and here is the place where you may find some glands if they're present in that organ. So as you're looking across different organs and trying to figure out what organ you're looking at, noticing differences in the mucosa and the submucosa will help you the most. As we move more superficial, here we have the muscularis externa. So this muscularis layer, you'll have two different layers of muscle running perpendicularly to each other in all of the organs, except when you look at the stomach, you'll actually have three layers. You'll add an oblique layer. But in most organs, you'll have two layers of muscle in the muscularis layer. And then the outermost layer is the serosa, and this will help to bind the organ in its place within the peritoneum. So when we look at this under the microscope, you'll be able to see these same four layers. So again, the mucosa is going to be the layer that's along the inside of the tube or lumen, and you'll see a lot of differences in the epithelium as you look at different organs. If you move a little bit more toward a more superficial layer, you have the submucosa, as seen right here. Then you'll see the muscularis. This all looks very smooth. You always have cells that are right next to each other. So you have smooth muscle here in the muscularis, running in two different directions. And then the outermost layer will be a thin layer of serosa. You should be able to identify all four of these layers in any organ that is part of the alimentary canal or GI tract. Here's just a quick image of the esophagus and a reminder that the esophagus is used for transporting food. It's not going to secrete any digestive enzymes. It's also responsible for peristalsis as you move it down, move that food down toward the stomach. Here's what the esophagus looks like under the microscope. So this whole layer right here is all part of the mucosa. You have your epithelium right here. Stratified squamous epithelium helps to provide protection against whatever you may have swallowed, even if it's a potato chip that you maybe didn't chew well, you have scratchy or pointy ends on it. You have that stratified squamous epithelium that helps to provide protection. You can scrape off a few layers and it's no big deal. Underneath the stratified squamous epithelium, you have the lamina propria. So there's our connective tissue that's just found in mucous membranes. And then you have the muscularis mucosa, just a really thin layer of muscle. And here is a submucosa. You may see some esophageal mucus glands here, but you may not depending on what section you're in. And then here we can see some smooth muscle. So this is the muscularis, and then out of this view would be the serosa. As we move on toward the stomach, you have different regions of the stomach that you're responsible for. So this area right where the stomach, right where the esophagus meets the stomach, this is going to be the cardiac region or the cardia of the stomach. This area right over here, this kind of hump that's farthest from the opening into the small intestine, this is the fundus or fundic region of the stomach. The main part of the stomach, most of this is going to be the body of the stomach. And then as we move toward the exit of the stomach, over here is the pyloric region or pyloric part of the stomach. This is also where you'll see the pyloric sphincter right here. It gates the way between the stomach and the small intestine. You also, not pictured here, but you do have a sphincter right here between the esophagus and the cardiac part of the stomach, and this is the cardiac sphincter, also called the lower esophageal sphincter. And so you have two gateways, one going into the stomach and one going out of the stomach. And you can notice here that there are 3 layers of muscle in the stomach. The stomach is unique in that way. This outer curve right here is called the greater curvature, and this smaller curve on the inside is the lesser curvature. This membrane right here that runs between the stomach and under the liver is the lesser omentum, and it is a peritoneal membrane. And here on the inside of the stomach, you can see all of these ridges, and some of them in the picture right here. These are the rugae of the stomach, all these ridges. They flatten out to provide more volume, more space available in the stomach. Here is a slide that shows the difference in epithelium as you move from the esophagus over here and into the stomach. Remember, they're all going to have the same 4 layers, but there is a pretty substantial change in the epithelium. As you look at the esophagus, you see stratified squamous epithelium, but as you move toward the stomach, right here, you see all these nuclei in a line. This is simple columnar epithelium. The cells are elongated, so that's the columnar shape, and simple means that there's just one layer. Compared to stratified squamous epithelium, many, many layers of thin, flat, squamous cells. We'll move on to the liver. Before we move on to the small intestine, we'll cover the organs that help to provide digestive juices that help the small intestine digest its food. So the liver is an accessory organ, and it is divided into lobes. So you have the right lobe here. When in doubt, remember the gallbladder is on the right side of the liver, right side of the body. So we have the right lobe, and then here is the left lobe. The right and left lobes are divided by this falciform ligament right here. And then extending out from there, you see the round ligament. If we turn the liver over, here we have the left lobe over here But as we move over here, we see two new lobes. So here's the caudate lobe. Caudate, think caudal. We know the vena cava is in the very back or posterior side of the body. So remember caudate and caudal, near the vena cava. Here, next to the gallbladder, we have the quadrate lobe. So this is the fourth one, quadrate lobe right here. And it looks even kind of like a quadrilateral a little bit. And then this would be the other side of the right lobe right here. So 4 lobes of the liver, and then you have the falciform ligament and round ligament. If we take a look at the microscopic view of the liver, you can see that you have lobules in the liver. So here's one lobule right here, and then you can see part of one lobule outlined right here in this picture. All of your lobules will have a central vein in the middle, and then on the edges, you'll find portal triads. And these portal triads will have a vein, may have a lymphatic vessel, and a branch of the hepatic artery. So you have those 3 things together make up your portal triad. So here is a real picture. Here's the central vein. Here is the outline of a lobule right here. All of that is a lobule. Here's another lobule right here, tracing the outline down here and up here. So that's another lobule. Here's a central vein right here, also a central vein right here. And then here on the outskirts, you can see a portal triad right there. As you zoom in, you can see the hepatocytes and Kupffer cells more clearly. So most of the liver will be made up of Kupffer cells, but you'll notice they almost look like cracks in the tissue. These are your sinusoids. So these are spaces for bile to drain. And you have Kupffer cells that are monitoring that bile. These are macrophages that can help phagocytose any debris or microbes if needed. The difference in how to identify these is Kupffer cells are frequently very dark and compact, and they are going to line the insides of sinusoids. So here's a Kupffer cell right here. Here's a Kupffer cell right here. Right here. Here's another one over here. And another one over here. So you see all these Kupffer cells are very darkly staining and compact compared to all of the other cells that are hepatocytes. So here and here, you can see lots of cytoplasm around them. The nucleus does not look dark all the way through like the Kupffer cells do. So those are some differences. Location, is it along a sinusoid or not? And size of the cell, do you see cytoplasm around it, or is it just this very darkly staining spot? That will help you tell the difference between hepatocytes and Kupffer cells. All of the bile that is made by the liver needs to drain down. Some of it is stored into the gallbladder, and the rest of it will be sent all the way down into the duodenum, and that bile will be used to help digest your lipids in the small intestine. So the duct system that helps to transport that bile is shown here. So here you have your hepatic ducts. We have a right hepatic duct right here, and this one would be your left hepatic duct. Where they join is the common hepatic duct. So this region right here would be the common hepatic duct. Your gallbladder, which stores bile, is going to send its bile, some of it, through the cystic duct right here and meet with the common hepatic duct. And after those two meet, this remaining duct is called the bile duct or common bile duct. And it's carrying bile from both the liver and the gallbladder, and it will carry it all the way down here into the hepatopancreatic ampulla, so hepato meaning liver, pancreatic referring to pancreas, and that bile will flow through the hepatopancreatic ampulla, through the sphincter, and here's your major duodenal papilla right here, and all that bile, well, not the entire contents of your gallbladder, but the bile that's sent down will all be expelled into the duodenum right here. In addition to bile, you may notice that the pancreatic duct is also sending its juices through this same common opening. So you'll have bile through the common bile duct and pancreatic juice through the pancreatic duct. They will all go through this hepatopancreatic ampulla, through that sphincter, and then out the major duodenal papilla into the small intestine. And so all of those juices can help mix with your chyme at that point and help digest it. Speaking of the pancreas, Here's your pancreatic duct running all the way through, all the way up to the tail. So here's the tail of the pancreas over here, and the body of the pancreas, and the head is this enlarged section right here. Pancreatic duct will drain all of the pancreatic juice being made by the acini of the pancreas. Here's the pancreas under the microscope. Most of it, as you can see, is acini. You do see the occasional pancreatic islet or islet of Langerhans. So pancreatic islets are responsible for secreting hormones such as insulin and glucagon, but these are relatively rare. Most of what you see in the pancreas is going to be these acini cells. You will see pancreatic ducts, and so these are just going to be round structures, simple cuboidal epithelium that will transport that pancreatic juice all the way down the pancreatic duct and give it to the small intestine. Here's the small intestine. It is composed of three main regions. You have the duodenum or duodenum, the jejunum, and the ileum. From the outside, you cannot see distinguishing characteristics between these three regions. And so usually what I'll do for a lab exam is help you figure out which section it is based on where I place the pointer. So if I'm asking you for the duodenum, if I say, name this region and organ, for duodenum, I'll put the pointer somewhere over here. And the large intestine is usually out of the way. And by its proximity to the stomach, you can see that I'm asking you for the duodenum. The jejunum I usually pin somewhere in the middle over here. And then the ileum, I'll make sure that I pin it right here, right where it is entering the large intestine. So you know that that is for sure ileum. So take care to note where your pointer is if you're being asked for the specific region of the small intestine. And then the contents of the small intestine will be moved into the large intestine, but it has to go through the ileocecal valve first. Ileo referring to ileum, the last part of the small intestine, and cecal referring to cecum, the first part of the large intestine. And here's your appendix right there attached to the cecum. But before we move on to the large intestine, I want to look at the small intestine under the microscope. So this is zoomed in on very, very far on one villus. So when you look at the small intestine, it is specialized for having a lot of surface area. And as part of increasing its surface area, you have these finger-like projections extending out of the mucosa. So you may see circles on the slide depending on how that villus has been cut, or you may see this long finger-like projection. So here's one villus that we've zoomed in on. You can see that the small intestine is composed of simple columnar epithelium. And so you can see all these nuclei in a row, and you can see that you have tall cells. So this is columnar cells. So simple columnar epithelium. You'll also notice that in an effort to increase surface area further, you may notice a fuzzy border or brush border. These are microvilli. And the microvilli are similar to villi, but they're just a whole lot smaller and they're on each cell. These are extensions of the plasma membrane and they make that border appear fuzzy. So there's your microvilli. On the inside of one villus, you'll have capillaries, blood capillaries, and also lacteals, which are lymphatic capillaries that are there in order to absorb lipids. Lipids are too big to get in blood capillaries, so that's why you have a lymphatic capillary, this lacteal, to absorb it. So that will be present inside each villus. You may also see some goblet cells, not a ton of them, but some. These are these white, puffy looking cells right here. They're usually quite round, and these will secrete mucus. When we look at each region of the small intestine under the microscope, you can see some differences microscopically that weren't evident before if you're looking from the outside on a model. So here you can see some of these villi extending out. So here's one, here's one, here's one, here's one. So these villi are all extending out. So all of this is going to be mucosa. All of this, all the way down here. This is the lumen, so this would be the inside of the tube. Here is the submucosa. Now this is where you're going to see the difference in the duodenum compared to other regions of the small intestine. You have duodenal glands right here, also called Brunner's glands. But these duodenal glands are only present in the duodenum. So look for these present in the submucosa. And then of course you have the other layers. Here's your muscularis and serosa. But when identifying the duodenum, make sure that you look for those duodenal glands. Here is the jejunum. It doesn't have any specialized glands available in it, other than the normal cells that are available in the small intestine. So here's the mucosa. You're still going to see some villi, and here you can see it looks almost like a finger that's been cut off, but that's just the way that the tissue is sectioned. So you will still see villi. All of this is still mucosa, all of that. You may be able to see some lacteals in there. Mucosa has nothing especially distinctive about it compared to the other two parts of the small intestine. And then here's the muscularis here and the serosa. In the ileum, it is noted that there are Peyer's patches in it. So Peyer's patches are part of your MALT, your mucosa-associated lymphatic tissue. So you've got secondary lymphatic organs, secondary lymphatic tissue that's available here in the ileum. So here you can see the villi. So this is kind of on the side compared to other slides that we've seen. So here is your mucosa, all of this, all right here. And then here are your Peyer's patches. They're quite large. They're not going to be all the way around the ileum usually. They'll just be on one side of the tissue. So if you're looking at a slide and you can't see the Peyer's patches over on the other side of the tissue, just follow the circle of that tube around the slide and see if you can find them on the other side, because the Peyer's patches are quite large. Make sure you're zoomed out enough. This is only 40x, but you should be able to see the Peyer's patches pretty clearly. And then you have your muscularis right here, and then this dark layer right here would be your serosa. So when you're looking at the ileum, look for the Peyer's patches. Moving on to the large intestine. So the large intestine, also called the colon, has several different regions to it. The first part, so coming out of the ileum, the chyme from the ileum will be expelled through the ileocecal valve and into this part of the small intestine, large intestine. Here is your cecum. This is kind of a blind pouch. Food doesn't really do much down here. It needs to go up. Before we go up, I want to point out the appendix right here. Usually seen on the posterior side of the cecum. That's where you're going to find that little tail-like appendix. But as that chyme moves into the large intestine, you're going to move up through the ascending colon and then across this right colic flexure, across the transverse colon right here, and then across the left colic flexure and move down the descending colon. This area of the colon that kind of looks like an S is the sigmoid colon, and then the remnants of your food will move down through your rectum and then finally into the anal canal. You'll notice that the large intestine is not a smooth tube like the small intestine. It has these little pouches on it. These pouches are called haustra. So you have haustral contractions that help in the final stages of digestion in the large intestine. You also have this line that lines the large intestine here. And these lines that you see right here are called Tenea coli. So there's how it's spelled right there, Tenea coli. And you can see it lines all down the length of the large intestine. Here you can see a little bit of the greater omentum. Normally this covers the front of the large intestine and the small intestine, but it's been reflected back in this picture. So you may see part of this greater omentum on a model or in your dissection. And this is just a peritoneal fold. So it's a fold in that peritoneal membrane, and usually there's some fat in it as well. So it just lays down on top of your intestines right there. It's a folded membrane. Under the microscope, here's what the large intestine looks like. It does not have villi and it does not have microvilli. So there's two big differences in the large intestine compared to the small intestine. So here is your mucosa right here, and it's folded up over here. The inside would be the lumen, or the inside of the tube. Instead of having villi that extend outward, you have intestinal crypts that extend inward. So you can see these intestinal crypts in large intestine. They extend downward into the large intestine. When you're looking at the mucosa, you may notice there are a lot more white, puffy cells. These are goblet cells. You still have simple columnar epithelium that is making up the majority of the mucosa, but you'll see a lot of these goblet cells. So if you're not sure if you're looking at large intestine or small intestine, one of the things you can look for is how many goblet cells do you see? What's the proportion of goblet cells to simple columnar epithelial cells? If you have a whole lot of goblet cells, that's a good indicator that you're looking at the large intestine. And then below the mucosa, we have the submucosa, this layer right here, extending all the way up here. And then we have the muscularis right here, and the serosa down here. Moving on to the respiratory system. So you have a couple of structures in the face that you'll need to identify. So you guys are probably familiar with the nose. You are probably familiar with the fact that you have a lot of cartilage present in your nose. So you have alar cartilage right here on the front, and then you just have bones up on this top part right here. If you look at a section of the nose, you'll notice that the nasal cavity has nasal concha in it. So these are large ridges that extend out. They're actually bones underneath the epithelium. And these help to provide turbulence to the air as you breathe it in. It causes the air to spiral, and so that air has more contact with the mucous membrane. So if you breathe in any debris, for example, it's more likely to get stuck in that mucous membrane. So you've got nasal concha that are present in here, superior, middle, and inferior nasal concha. And then you can see that the nasal cavity and the oral cavity down here are actually connected down here. So the region leading into the pharynx is shared by both the nasal cavity and the oral cavity. And then down here you can see just a little bit, here's your esophagus back here and your larynx right here. The epiglottis helps you to make sure that air moves down your larynx and then ultimately to your lungs, and food goes down the esophagus. If we take a closer look at the larynx, here is that epiglottis we were looking at, and his cartilage underneath. So here's a front or anterior view. Here's your cartilage. This is the hyoid bone. And then here's a posterior view. From the side, your epiglottis looks kind of like a little flap or a leaf. And that is what closes to make sure that air goes down into your lungs and food moves into your esophagus. In the front of the larynx, you can see a couple of very large pieces of cartilage. You have thyroid cartilage right here, and then you have your cricoid cartilage right here. And then moving on down from here, you start the trachea, and these are tracheal rings, tracheal cartilage. One interesting thing about your tracheal cartilage is it doesn't go all the way around the tube. It's C-shaped, so it goes most of the way around, but there is a section here without cartilage. If you look at this cricoid cartilage from the posterior side, you can see it's quite a bit larger on the posterior side. On the inside of this cartilage, this protective area, you have vocal folds. So you have a vestibular fold right here, it's a superior layer, and then you have a vocal cords or true vocal folds right here. So your vocal cords or your vocal folds are what will vibrate and what cause you to speak and sing. The vestibular folds are your false vocal cords. So here's your larynx and your laryngeal cartilages. Moving on down, we have your trachea. So you've got tracheal rings made of cartilage that extend most of the way around the tube. And then you start to see a lot of branching. So you have your primary bronchi that extend right and left that are going to serve your right and left lungs. And beyond that, you'll see a lot more branching. As you move down your bronchi into smaller and smaller bronchioles, this cartilage will start to disappear more and more until it's eventually gone. If we look at the trachea under the microscope, you can see some of that cartilage right here. cartilage is hyaline cartilage, and to me it just kind of looks like eyeballs, lots of purple stain here. So hyaline cartilage is here. Here is submucosa. So we're not in the alimentary canal anymore. You're not going to have all of the same layers that we were looking at earlier, but you do still have a mucosa and a submucosa when we're looking in the trachea. So here's your mucosa. You have an epithelium, you have a lamina propria down here. Here's your submucosa, where you may see some seromucous glands right here. You won't see them in every part of the trachea you look at, so you may have to scroll around a little bit before you find them. And then deeper you have this hyaline cartilage. One thing that I want to point out about your trachea is that this epithelium right here is pseudostratified ciliated columnar epithelium. So pseudostratified means that it looks like it's stratified, but it's really not. You just have nuclei that are kind of all over the place, and so it gives the appearance of being stratified. And it is also ciliated. So you have, it looks like a fuzzy border right on the edge here by the lumen, and that is due to the presence of cilia. Cilia and microvilli are different, although they look very similar under the microscope. Cilia are actually small hairs or small tubules that are put in the plasma membrane, compared to microvilli, which are extensions of the plasma membrane itself. However, under the microscope, they do look very similar. They look like a fuzzy border along that epithelium. Here are the lungs. So here you can see the trachea and then your bronchi leading into the lungs. The lungs each are divided into lobes. So here's your right lung right here, and your left lung is here. Your right lung has 3 lobes. So you have a superior lobe, a middle lobe, and an inferior lobe. The superior lobe and middle lobe are divided by a horizontal fissure right here. And the middle lobe and the inferior lobe are divided by an oblique fissure. On the left side of the left lung, you only have 2 lobes. So you have the superior lobe right here, and then an oblique fissure that divides it from the inferior lobe. Another feature of the left lung is you have this cardiac impression or cardiac notch right here. Because the heart kind of tilts to the left, it's taking up some space where the left lung would be, and so you have a little bit of an indentation here called the cardiac notch or cardiac impression. If you take a look on the medial view of the lung, you can see the hilum where all of the arteries enter and the veins exit, and you've got your lobar bronchi that are all entering here. And this is just showing you as you move down your bronchial tree, you've got plates of cartilage that are slowly disappearing. So you started with rings, and then it slowly starts to disappear a little bit. So you have plates, and eventually those plates get smaller and smaller, and eventually they disappear altogether. So here's your bronchiole. This is one of your smallest respiratory passages. And instead of cartilage, you have smooth muscle that's replaced in here. And this smooth muscle, just like any smooth muscle, is capable of constriction and dilation. And so that will help regulate airflow into your lungs. As you continue to move down, you have a little bit more branching. Here's your terminal bronchiole that leads into a respiratory bronchiole, and respiratory bronchioles will lead into an alveolar duct, and then finally your alveolar sacs. Here's a little bit of a closer look at your respiratory membrane. When we look at the slide, you'll be responsible for identifying pulmonary arteriole, this terminal bronchiole, and then the alveolar ducts that move into the alveolar sacs. So here's a picture of what that looks like. All of these tiny little circles are individual alveoli. Singular would be alveolus. But sometimes you can kind of see the outline of some of these alveolar sacs. So here's one right here. Here's another one right here. It almost looks like a cluster of grapes that you've cut open and looked at the middle of it. Here's another one right here. When you're looking for bronchioles, this picture is a little blurry, but you can see just a little bit of a wavy border right here. So the terminal bronchioles have a little bit of a wavy border, and so that's how you can identify them from pulmonary arterioles. Arterioles will have a very round inside, and so you can see another one over here. But bronchial, as you can see a little better in this one, they actually have a wavy border, and so that will help you identify bronchioles compared to arterioles. And sometimes, if you're lucky, you'll be able to see the erythrocytes on the inside of the arteriole that will help you identify them. Here's another picture showing alveolar sacs and alveolar ducts. So here is the outline of an alveolar sac. Alveolar ducts are the pathways that lead into the sacs. So here's another one. Here's an alveolar sac over here. Here is an alveolar duct right here. And here you can see that bronchiole a little bit more clearly right here. And compare that with a pulmonary arteriole, much rounder. There are a couple of muscles in the respiratory system that are important for providing the pressure differences so you can get inspiration and expiration. The two main muscles that you need to know are the diaphragm right here, this dome-shaped muscle here, and then your external intercostals located along the outside of your ribs here. So your external intercostals and your diaphragm provide all of the muscle power that's needed for you to inhale. And then when you exhale, all you do is relax those muscles. Now for forced expirations, you'll use some of these other accessory respiratory muscles. But just when you're sitting quietly, a normal quiet breath is accomplished just by your diaphragm and your external intercostals. So make sure that you identify these on the models in lab. Moving on to the lymphatic system. When you look in the mouth, you have several different tonsils that are there. You've got a lingual tonsil near the tongue, palatine tonsil near the soft palate right here, and pharyngeal tonsils way up here toward the nasal cavity, also called your adenoids. If you take a look at a tonsil under the microscope, this is what it looks like. So you see these large circular lymphoid follicles. And another indicator of the tonsil is tonsillar crypt. So you'll find this looks like white space that's extending into toward the middle of the tonsil. So tonsillar crypts and lymphoid follicles will be the round areas. Lots of leukocytes, B cells, and T cells will be here. In a lymph node, here's a picture of what one looks like. You'll also note that you have these rounded areas right here, lymphatic nodules here. However, in a lymph node, they're going to be clustered around the outside or around the cortex. So when you look at it under the microscope, you see the same lymphoid follicles that we saw in the tonsil, and so you may have some confusion differentiating between the two. But lymphoid follicles in the lymph node will always be around the cortex along the outside. And then it's also a little bit clearer in the lymph node, the difference between the cortex, this outer layer, all of this right here, and the medulla, where you have all these little sinusoids and trabeculae. So here's the lighter staining medulla here, and then all of this is all cortex. And you'll find the lymphoid follicles always in the cortex. Here's the spleen. Here it is in a person. It's going to be very close to the pancreas and above your left kidney. When you look at the spleen under the microscope, most of it is going to be composed of red pulp. There'll be a lot of erythrocytes, but you will also have regions of white pulp where you'll find leukocytes. And these tend to stain a little bit more purple. They're also circular in area, similar to the lymphoid follicles, but this is where all of your leukocytes would be in this white pulp. So think white pulp, leukocytes, because they have granules and nucleus, it takes up a lot of stain. Even though it's called white pulp, a lot of times it stains a pretty dark purple. Most of the spleen will still be composed of red pulp. The circles will be the white pulp. So that's it for our overview for your human digestive, respiratory, and lymphatic systems lab. Don't be afraid to ask questions in lab. We are happy to help you. We know the histology is a little bit tough. Try to look for the differences in the epithelium and the digestive system, and just take notes of the differences between all of the organs that appear similar to you. So please feel free to come to any of us for help anytime you need it.